Great, thanks. Thanks to the organizers for the invitation to speak, and thanks to you all for hanging around for the last talk of the day. I made the assumption that it was probably not going to be in everyone's best interest to have a deep background on FFPE and all the challenges. I think that's probably been gone over very, very well so far today. So I'll just kind of hit a couple highlights as far as from an application perspective of some of the things we've looked at. But prior to that, I'll have just a few introductory slides of the lab and the projects and some of the perspectives that we've taken particularly over the last 3 months or 6 months for this Multisite Research Alliance that I'll talk into a little bit of detail. So probably like many places, as sequencing capabilities have gone up, so has a variety of both yield and numbers of samples that have been enabled as the costs have gone down. So this is just a vignette from our lab through 2018 in terms of numbers of both yield and bases, and then transcriptomes, whole genome, and exomes as far as amounts. And then over the most recently, we've actually gotten rid of all of our, we've condensed the sequencing platforms down to NovaSeek, as well as, and the main motivation of that was really to try to streamline the reagent platforms, having between HiSeek 2000, 2500, 4000, HiSeek X, et cetera, et cetera. Everybody's reagents were different. It was logistically fairly challenging. The benefits were, of course, the 8-lane formats, which you have some of the considerations, and now these 2- and 4-lane formats. But going from the complex reagent stream and then looking at the NovaSeeks in general, we decided to take a little bit of a chance on the early days when we started setting it up and decide to essentially get rid of all the other platforms and condense everything on the NovaSeek that we affectionately like to refer to now, because it's ease of use to streamline things in the lab a lot. It's like the Keurig of sequencers. Like you just literally put the cartridges in and press go for the most part. And so we have that, plus ISeek, as far as we use it primarily for QC for this platform, and I'll mention that in a little bit of detail. And then what turns out to be a very good balance to kind of round out the output capabilities is NextSeek and MySeek. So 2 of each of those, and then 10 NovaSeeks is kind of what the platforms look like overall. So the lab looks at a wide diversity of projects, from physiological measurements for performance, neurological disease, cancer, and then some more global population efforts, and then some of the projects I know that I'm sure hear about in the keynote, as well as many people in the room and at the meeting I've worked on with Chris Mason, the MetaSub project. We've done quite a bit of sequencing for that. And then on the oncology side, this is a very recent Circos plot from Cosmic, and again showing, of course, the genome and then the various types and areas of mutations. And what it boils all down to is, and it's really no surprise, is that cancer is a complex disorder. But interestingly, and as a reminder, when we think about predisposition testing, that only 10% of cancers have a germline mutation that has an attribute towards increased risk. Right? So it's in the minority of cases, but 80% have somatic mutations that do not appear in the same germline samples, and then about 10% or so have both. I'm obviously painting with a bit of a broad brush, but it's to illustrate, that this is a genome-wide phenomenon, and that somatic mutations and the ability to measure transcriptional and DNA changes are fundamentally important for the continued understanding or continued advancement of the understanding of cancer. So 3 project areas, and I'll focus on this one in the upper right-hand corner, which actually this should say is supporting 19 now national cancer centers via a unified molecular platform for the analysis of adult cancer. We've done quite a bit of work with St. Jude on a large longitudinal study of whole genome sequencing, and this is now approaching 10,000 genomes sequenced. And then we're a sequencing center for the Gabriela Miller Kids First, which has a pediatric cancer component as well. So most of the discussion and data I'll focus on this, and then interestingly, to give the differentiation between adult versus pediatric cancer, this is a figure out of a recent paper published that St. Jude published on the behalf of the work that we did together. And what this is showing is when you think of pediatric cancers, there is a clear benefit for genome sequencing or exome sequencing, and then lifelong genetic counseling. Because what this is showing is for genes that were found to have a mutation, and these are the genes shown around here. The size of the circle is the number of times that gene was mutated in that cohort that I mentioned, and then the pie graph on the inside is the type of secondary or tertiary cancer event that those patients had either or had been diagnosed with. And so what it's showing is on essentially looking at childhood cancer survivors which is the cohort in St. Jude Life and by extension the childhood cancer survivorship study, that somebody with a BRCA1 and 2 mutation has, of course, a high probability for breast, but also a significant risk for thyroid and sarcoma cancer. So the idea is with genetic counseling on these survivors, you can have appropriate monitoring and appropriate, early interventional programs for, of these wide variety of different cancers, depending on what mutations they had. And so some things were quite deterministic, as you see over here, with, mangioma or non-melanoma skin cancers or even sarcomas. So on the pediatric side, very, very clear value in looking at the underlying genetic predispositions. But as I mentioned in the earlier slide, you see certainly some value in the adult cancers, but again, probably not quite as great. So the goal of the ORIEN program is fairly simple in concept. So it is getting samples from this collection of national cancer centers. Many of them I'm sure are very familiar, and I'm sure many of them, there are people from one or several of these in the audience. So it's essentially founded out of Moffitt with the James at Ohio State, and then Huntsman and UNC Norris have been some of the larger centers contributing so far. These did not start all at the same time, so there's a variable number of samples from these sites, but the idea is a unified molecular platform to analyze a quite high diversity of tumor and matched normal samples from patients. And basically, it's deep exome sequencing on the tumor, about half the coverage on the normal tissue, and then RNA sequencing on the tumor as well. And I'll get into the details a little bit more. And then the overall program, if it's of interest to anyone, is actually quite well described at orioncancer.org. You can get a bit more in terms of, there's even some funding opportunities and announcements through that group. It's managed by a company called Emgen, which is a spin out of Moffitt. And basically the goal is to have both a new kind of research alliance, very efficient mechanism for data sharing to guide discovery, and then even advancing precision-based therapy. So some of the funders of this are a variety of pharmaceutical companies that are hoping to translate this into either matching patients up to the right clinical trials or in fact more advanced drug development and treatment developments from this data. So it's a fairly large effort. And one of the early slides from it is here. So this is looking at samples from different centers. So this was a bit of a, between Moffitt, OSU, Norris, Huntsman, and Rutgers. You can see the vast majority at this cut, this is actually probably about 8 or 10 months old now, was the majority from Moffitt and OSU, as I mentioned earlier. But then a wide, pretty broad diversity, as you can see by this pie chart, and obviously everything's not represented in the colors here, but a wide diversity of cancers in tumor types, and they're kind of listed over here in terms of numbers. So not surprising, this number is inflated because this is where the matched normal often comes from. And then you can see all the way down to some relatively rare numbers of bone and uterus, adrenal gland, et cetera. And then there's a kind of a miscellaneous catch-all. Like when the samples were accessioned, if there wasn't a specific tumor site that was annotated, it goes into this bucket until it's annotated later. Occasionally, there's a disconnect between when samples come in de-identified and then when the clinical information follows. So we kind of have a catch-all for some of that until the data catches up with the sample. So what's done as far as the genomic platforms? RNASeek it's primarily an RNA access program, and the earlier talk noting the diversity or the complexity limitation this had from a transcriptional perspective, I think we're right on. In this particular case, the main focus is on detection of fusions and looking actually at the mutational profile seen in the RNA transcripts, not necessarily a transcriptional difference between the samples, although that's not ignored, that's just not the primary goal. We sequence that at 100 million paired reads per sample, so fairly deep sequencing. It means a 96-well plate per S4 flow cell. For the exomes, germline is 20 gig data per sample, so that's one S4 flow cell per 96 samples, and the tumors are double that, 40 gig. So it really works out to be 150, 200x on the germline and close to 400x on the tumor. One of the main reasons this is done is the variability between samples, the variability on those qualities, is by sequencing at this depth and really never having to do top-up sequencing. So some will be well overcovered, but almost everything always hits passing metric first time through. Turns out, just because of the volume of samples going through, very, very handy. But these are the specific sequencing conditions. These are at pair, and everything's at paired 100 as far as actual read lengths. We also do some panel work, TST 170, TSO 500, and then there's actually a custom RNA DNA platform which mirrors the goals of this. So simultaneous DNA and RNA isolation out of a tumor sample, and then those RNA and DNA samples both go through a panel that, again, is same spirit of these from an overall workflow perspective, but a completely different platform. And it's not quite ready for prime time. And then on the whole genome sequencing side, we've done a variety of whole genome sequencing from FFPE. I'll mention some of that in a moment. Circulating tumor DNA, some single cell work. Obviously, the coverage limitations and some of the challenge with these samples are well recognized. And again, I'm not going to go too much into this data. And then finally, long read sequencing. Trying to tease out much more of the genomic and genetic complexities within cancer that are clearly driven by copy number variants, which are actually, I think, becoming increasingly well recognized as important. And then the last thing about this slide is this is certainly from most mature to least mature as far as platform, and in fact, most highest number of samples to lowest number of samples in terms of the workflows. So as far as considerations for FFPE tissue processing, again, I'm sure I know the challenges have been very well represented today, and so these are more of, I think, some specific considerations that we've found to be very, very helpful. Some of them are very obvious. Maybe some of them will be helpful, so I'd kind of put them up and go through them quite quickly. That, not surprising, this is for post-surgical resection. This is really on the pathology side. This isn't the handling of already fixed tissue, but tissues really are optimally fixed within an hour of recovery. That has a significant influence on overall utility downstream from that, particularly for genetic studies. Optimal fixation time is between 12 and 24 hours, depending on tissue type. This can be important. In some of this work, we've found that there are cohorts of tissues that if the surgeries were on Thursdays or Fridays, sometimes they were allowed to fix over the entire weekend. And those are significantly different than tissues that were resected during the week and only fixed for, again, overnight or for 24 hours. And the other absolutely critical part, again, probably obvious to most, is that formalin that is at a neutral pH really has to be used. If it's an acidic formalin, it just destroys the tissue. So these are some of the things. We have I guess, a more formal description of some of this that we do provide to pathology labs and surgical centers, et cetera, to try to help with some of this. These types of changes have been actually pretty well received by some of the larger pathology groups that we've worked with. So that's been good. Fixed tissues really have to be completely dry before embedding. That makes another big difference in the longevity of that material. And then what we found, and I heard some of the questions after the last talk, is that storage conditions rather than age are the largest determinant in utility of that tissue downstream. So we've worked with stuff that's 30 and 40 years old. If the blocks were large enough that we could section away the oxidized outer layers and there was enough tumor cellularity, those work just as well as a 1-year-old or 2-year-old sample. And it really comes down to these considerations rather than age, I think is more important. And the storage of blocks without cut faces prevents unnecessary exposure and other environmental factors that damage DNA and RNA downstream. So if you can get to a fresh part of the block, it actually works really, really well. Tissue thicknesses between 10 and 20 micron with a surface area between 50 and 300 millimeters square, right, ballpark. And that's just to get enough material to isolate. And then excess paraffin, of course, has to be removed before any purification steps. A couple different ways to do that. A couple quick mentions that we found to be pretty effective working with a diversity of collection sites is of keeping section samples cold, and if they can't be delivered cold and delivered within 24 hours of sectioning, that they get put under mineral oil, and that just prevents all those oxidation effects. And then, of course, the mineral oil has to be removed, but the mineral oil will also play a role in the deparaffinization of the sample as well. So it's a little bit of a win-win, but there is the hassle of pulling off the mineral oil. And then, again, I know this was probably covered well, but these are all the, without going into any specific, these are all the, normal DNA damage modifications that are seen in these types of samples. And so what one mechanism we're looking at now and on these projects, and I don't have a lot of significant data on, is really just running since these are short insert libraries already, then we're running those on longer read sequencing conditions to get essentially both strands, so intentionally overlapping the strands with the paired end reads, so that those, a number of the, the C to T transitions, et cetera, that happen, you can actually correct for them because they're only detected on one strand, not both. So if you have the paired reads, you can very, very robustly tell them apart. So you can take a 250 or 300 base pair insert library and run it at paired 250, and again, intentionally, run those in either direction. So we've looked at that on both the ISeek as well as the NovaSeek. Works pretty well, and some of the early testing stuff we looked at was just to overall make sure the base quality stayed high on the end of the reads to make this a viable approach. And since you are sequencing, a bit of a head to tail sort of relationship, on one read or on one direction when the read quality is going down, of course on the other read that's where your read quality is highest, so it works out pretty well. So that's an example of ISeek. In this case, it's an exemplary library of just microbiome. This is actually the ISeek 500 cycle, so going out, similar thing. You see similar patterns on Chromium and RNASeek libraries on the NovaSeek. And if I kind of go to the punchline where we, again, so kept the insert sizes relatively short, here in the 300 base pair range for a normal in tumor, and this was fresh frozen, this was FFPE, and of course this was cells or out of a cell line, so it was control. And then looked at the mean coverages and some of the usual coverage statistics, and this was, again, intentionally overlapping. And so in the FFPE samples, you can pretty accurately quantitate that rate of sequencing-based or artifactual errors in the mutational calls, correct for them, and one of the ways, I don't have the data completely prepared yet, but one of the ways we've done that is looked at that first from a variant calling perspective, actually made the correction in the FASTQ files or on a separate copy of the FASTQ files, and then run them back through variant calling to try to see how much that actually influences it. And not surprisingly, it's highly dependent on the sample and highly dependent on, so it's tough to draw any broad conclusions other than to say this overall scheme, I think can be reasonably effective and can be reasonably empowering if you're willing to essentially pay for intentionally duplicating your read coverage and then designing the downstream pipeline to utilize that information rather than throw it out. So it does require some careful modifications to a typical pipeline. So in that spirit, again, sequencing quality remains pretty high on the genomes. We're seeing easily into the 90s and mid 90% at Q30. And you can just see that slight drop off at the end. But again, since you're doing the head to tail, it's low quality lines up with high quality on the other side, so it works out pretty well. And this is another example there. So what that's led to is, as we looked at this kind of from a holistic perspective of saying we can kind of do more sequencing and get more depth of coverage to correct for some of these problems. We can overlap reads to correct for some of the problems, and then we can also add more automation steps, which we've transitioned most of the automation steps to Beckman I-series robots, both i5s and i7s. And most of the library prep that we use, or all the library prep we use for the exome work, is all based on it's a mildly modified version of the Kappa hyperprep. So it's a end repair add atypical considerations. There's the Covaris shearing on the front end, followed by ligation with dual unique indexed adapters, which of course is very important. One of the things we also do in the dual uniques on this number of samples is we mix up the pairings. So we have 192 true dual uniques, but then that makes, right now we have about 40 sets of those where we just switch up the pairs. And all that gives you is an ability to tell flow cell 1 from flow cell 2 to flow cell 40, right? It doesn't actually give you more dual uniques to work with, but it just gives a traceability in the laboratory that you can have unique patterns of indexes on each plate as they go through processing, which, again, it's a extra traceability step, QC check, et cetera. And then since we also run a clinical laboratory, the last CAP inspection we did, they were very, very positive of that sort of workflow. So just as an FYI, if it's helpful. Again, all we did was switch up the pairings. You do have to do the validation runs on the pairings because there are certain pairs of indexes that all of a sudden will change behavior. So I think we've thrown out now about 8 different pairs over the set of 192 and redesigned, because when we switched up the pairs, the performance changed. So it is something to keep in mind. Conclusion is switching up index pairs can have an effect on how well they're represented downstream from that. We're now using the ISeek for most of the QC in place of qPCR here. And then it's a normalization, pooling, sequencing run. We can adjust pooling if necessary and onto sequencing run too. And this is obviously very easy to do for the tumor samples where there's 2 flow cells, and that's exactly the workflow. For the germline samples, we'll occasionally, it varies depending on batch sizes, we'll occasionally take that 192, make one pool, do a flow cell and a flow cell, so we can do that as well. But it's also not unusual that we'll do one single run for the germline samples, which is why the data off of the ISeek turns out to be very informative because we can adjust pooling before going to the production run, which again works very, very well. So, if you're running at least 96 samples on this and you're buying these from Illumina in the 4-packs, it's in our hands at least, it's pretty equal to cost per sample as Kappa. I think everybody's mileage may vary a little bit there depending on how you do costing and kind of depending on some of the other considerations. So as we've looked at this holistic protocol, we kind of we really wanted to look at optimization of the full genomics workflow. The improvements I've been mentioning along the sequencing side and the informatic approaches have drastically improved the analysis of cancer samples. Certainly not unique to our group, but I'd say in the field in general, certainly as evidenced in the last talk that talked about the different pipelines and their optimizations and how it can. But can we amplify those improvements by taking a very basic concept, right? If you really looked at carefully improving the sample quality and the yields, how much of that you can then amplify any optimizations you have after that if you have just better quality samples. And so this was driven by an observation for a substantial number of the ORIEN samples on the RNASeek side, we were getting somewhere around 5 to 20%, depending on how we divided the cohorts of samples that had a DV200 metric of greater than 20%. And for RNA access protocols or even RNA sequencing in general, those longer fragments and getting a good percentage of the fragments longer than 200 base pairs is pretty important. And it is, in fact, almost a gating mechanism for RNA access for success. If the DV200 is below 20, the RNA access protocol fails. It's a coin flip whether the sample works or not. So we really, really wanted to improve RNA quality. Yields were pretty decent. We just didn't want to lose yield. And so improving the yield wasn't an effect. And so in the interest of time, and just kind of fast forwarding without going through a lot of data of head-to-head comparisons, we tested a wide variety of isolation protocols, all the usual vendors and all the usual protocols, and really settled on a protocol that has the Covaris, the AFA technology as the primary step, both here and then again here, for essentially a paraffin removal and emulsification, in which is really a bit of a lysis of the cells as well, and then a more, another disruption to essentially get the DNA out. And so this is an RNA-DNA workflow, as you see here, RNA-containing soup and a DNA-containing tissue pellet. And this is a figure from Covaris showing the, actually from their protocol, but it gives you a nice flow chart. And then we've exclusively used the bead-based protocol. Rather, they have a column-based and a bead-based protocol. This is exclusively the bead-based protocol. What's not shown on here, which we found to be quite critical in the success of this, is the use of DNase or RNase, respectively, to degrade away the nucleic acid you don't want. We found we needed to actually about double the DNase treatment, both in terms of amount of DNase and time to really get rid of the DNA out of the RNA samples. RNase treatment by protocol was fine. So that is one, I think, note to keep in mind, that the amount of DNA hanging around was somewhat significant. It's also been variable per sample. A lot of it probably has to do with how well the cells are lysing in that first stage, and how well the DNA is pelleting when you make this tissue pellet. And then the backside automation for this particular workflow is a Kingfisher. The really simple reason why is we keep the other robotics busy on more complicated protocols. The necessary steps here are actually pretty simple from a plate handling perspective, and this $50,000 or $60,000 robot fits that need really quite well. Easy to scale up and down, does a good job, and so that's how we've automated the backside of this, the bead handling step and the rest of the purification steps. And so in the early part, so from January to October of last year, we did about 14,000 DNA and RNA samples from the ORIEN project. This is how they break down in terms of amounts, in terms of DNA and RNA. So you see on the RNA extraction, it was about 5,000. DNA, not surprising because of the normal samples that are up here. These are more closely balanced. We don't always get a normal for every tumor. We don't, and in fact, in some cases, The tumor and normal samples are discontinuous with each other, again, when they come in. So these don't always line up on an exact one-to-one basis. That actually turns out to be a reasonably helpful QC step at the very, very end of the process, because then the normal and matched tumor are separated in batch and separated in time. And then, of course, there's always a check to make sure their genotypes align and everything else. So it works out pretty well. But the bottom line being, it hasn't been dozens of samples. It's been thousands as far as the remaining data that I'll show you in some of the FFPE samples. And then we're currently processing at a rate of 400 to 600 samples a month. And that's been ongoing since October and accelerating a little bit now that we've got the isolation stuff worked out. So these are the typical yields and amounts that we use into the various downstream processing. And I won't belabor it. The point of this is say they're standard. They're not doing anything special in terms of amounts, nothing ultra low input and nothing using a ton of material. What's probably more important are the sample requirements that we found that are ideal for the extractions. Blood, again, pretty standard, 1 mil to 2 mil. For frozen tissues, it's this 50 to 80 milligram range is great. If you go much over that, you kind of overload the larger size tubes from Covaris. So kind of this 50 to 80 milligram range is really, really good. On the FFPE, assuming excess paraffin is removed, these 3 sections 10 micron thick. If it's slides, 5 slides, and if it's cores, 3 cores at 1.5 millimeter diameter, 5 millimeters long. So again, looking at hundreds to thousands of samples, this has been, I think, a really good sweet spot for tissue requirements. And interestingly, the curls or the FFPE sections are actually the lowest quality, lowest yielding of them all. The slide scrapings work great. The cores are by far the preferred if they're available. And again, in terms of when we look at yields and overall qualities. So these are typical yields. Again, blood, 10 to 15 micrograms of DNA, and if it's a packaging tube out of RNA, 3 to 7 micrograms, again, standard protocol. So we're typically seeing 5 to 15 micrograms out of fresh frozen, 2 to 6 out of FFP, and then again, the cells. We do occasionally receive sorted cells, so if it's 10 to the 7 cells, 8 to 10. So as you see here, plenty of material to work with overall, plenty to QC, and plenty to work with in the downstream assays. In fact, even to be able to do multiple assays, since you're at multiple micrograms for each sample, and that's assuming these conditions. Importantly, remember the earlier comments about the handling. So the time from sectioning to processing, et cetera, is actually quite important and clearly will influence some of these things. So since all of our earlier work was done with All-Prep from QIAGEN, the first thing we did was lined up a DV200 comparison between some samples that we had already prepped on QIAGEN and then re-prepped on Covaris. And these are the tumor types that are shown along the bottom. As you see, your ovarian, breast, et cetera. And for the most part, with some relationship to tissue source, we'd see or in all cases, an increase in DV200 from what's shown in red, which is the Covaris protocol, and then shown in blue is the QIAGEN protocol. And as I mentioned, this was had a relatively high failure rate of samples going into RNA access with the Covaris protocol. So this was the most significant change we were hoping to make. Now, the overall method, it is still quite manual, but that is the nature of the samples, right? These are FFP samples. They have to be shipped some way. They're curls or cores. I mentioned all that variability. So there is an important logistical or at least lab consideration. So this is just some pictures I grabbed on Friday showing how we queue up both the tubes that are used for the focused acoustic AFA. I'll just say AFA. For the AFA work, where the tissue goes into the tube. All the tubes are 2D barcoded, so they're coded from Covaris 2D barcoded, but it's always they're labeled by hand in ink or in Sharpie, so they're human readable as well. Importantly, you can't put a sticky label on, right? You can't put it on the glass because it influences the process of the machine. And then if you try to find somewhere on the tube, you either can't read the barcode anymore, or you end up with a label that goes across where you actually take the screw top off. So they're just labeled by hand. We haven't figured out a better way to do it. If anybody has any ideas, I'm all ears. And then these are the downstream tubes again that do go onto the robotics. These are all barcoded, which are read as they go onto the robotics, and they're also just again human readable, made on the top just to make things easy. So we have a pretty good system as to how things are lined up. It's a bit of a production line now, but I just at least wanted to mention that between this and a small amount of specialized equipment, both a heat block that holds those tubes quite specifically, as you see here. So this is a great version of that. You need a centrifuge that holds the tubes as well, so you can spin down that tissue pellet that's mentioned in the protocol. And then we use an absolute ton of the multi-channel pipetters, especially the expandable ones, which you see in these here, and then or these 4 that are here, to actually just make some of that liquid handling steps a lot more convenient. And so in the in these racks, the expandable pipettes can reach across this direction, so it works out all right. So what does it look like as a punchline? So the optimized protocol, you can see, here's the old way, the standard protocol. 22 out of the 95 samples had a DV200 of greater than 20%. You can see the ones that fall over this line. And then in this case, this is sections, so these are the curls. Pretty dramatic improvement. There was only one sample that fell below, so 89 out of 90 had a DV200 of greater than 20%, with, in fact, most of them quite a bit higher. Really, almost the majority is over 40%. So a dramatic improvement. Obviously, these longer RNA transcripts should yield better representation of the transcripts and potentially less bias in the downstream analysis, and of course, certainly a much much-higher success rate through the overall protocol. Even more dramatic improvement in cores, which is why I mentioned that they're more preferred. If you actually look at the 40% line, there's really nothing even below 40%, with some of them even touching above 80%. Now, the first time the senior scientist in the lab, Nirpesh Prasad, showed me the data, I thought, well, there's absolutely no way this is real. So run it again on another set of samples and run it again on another set of samples. And this exact pattern continues to repeat. So it is a dramatic change in the cores, which is why we've now gone to, cores are now the preferred rather than curls. Now, whether or not this was, it almost seems as the reverse was true for the QIAGEN protocol, the standard protocol, that the cores performed less well than the slides, but at least again in this case, clearly a dramatic improvement. This is all on the RNA side. If we look at DNA, yields are good. The improvement on the DNA side is modest to insignificant. But it's, so this is again clearly an RNA improvement. I just want to make sure there wasn't the expectation that you'd all of a sudden see multi-kilobase to megabase lengths of DNA coming out of FFPE samples, because that's not what's being observed. But it is a clear, significant improvement on the RNA side. And this is certainly of interest and important when we look at things like the TST panel, where these fusions and splice variants from RNA, but the longer the RNA species, the more accurately these are detected. And when we look at this, again, in a separate set of lung cancer samples, lung cancer FFPE material that was quite a bit older, quite a bit lower tumor cellularity for the most part, and we pulled 240 FFPE blocks. 202 of those samples had a DV200 of greater than 20%, so the vast majority, so a bit lower. So I think this is another example of presenting a more of a real-world effect. So we had those, several hundred of that were mostly breast, ovarian, and some other cancer types. This was a specific set focusing entirely on lung, but we did still see about an 85% combined sample plus library pass rate for the RNA processing. So still quite good, but again, it's pretty good overall. And so now with the TST-170 processing that these were run on, this has been automated Beckman automated this on their robots, and there is a very nice capability of doing up to 32 samples on an i5 span 8 simultaneously, and that is the RNA and DNA workflow. It's a 2 to 3 day workflow total, and it's automated to the extent of you unpack the Illumina kit, right? Load the individual tubes on the robot deck, load the plastics, load the samples, and press go. Like there's no master mix making, there's no incubations off deck. It's truly a canned protocol. Beckman did all of that development, and we've been incorporating it because we wanted to have the reason we wanted this particular prep to be fully canned was so that we wanted to be able to have fast turnaround time, multiple shifts of technicians being able to do it and have it run exactly the same way every time, no matter who was doing it. So that's why this was done that way. To be fair, if you run it manually, right? If you use a hybrid approach, some automation, some manual, with a really experienced technician, you will see slightly better libraries out of the manual protocol than out of the fully automated. But out of the automated protocol, you'll get exactly the same. We see our control samples are extraordinarily tight CVs from run to run to run to run to run. So it kind of depends on what your goal is. Is it a highly reproducible process or one that you're extracting every bit of quality out of that? It's somewhat expensive for large-scale work. That has nothing to do with the automation schemes. It's just that I think the Illumina cost for that kit, for the number of genes in the kit, it remains pretty expensive. You sequence the DNA deeper than the RNA, and so this is just an example of just showing the quick check on the indexing, and this is pretty typical for an indexing QC on that. So this is this is the DNA side, and then from the same samples, the RNA side, just to give you an idea of that differentiation. So overall results. What we found is RNA samples with DV200 scores greater than 20% increased from 15 to 20% on that old protocol to over 90%. We see a higher quality out of cores than curls. Slide scrapings are actually quite similar to cores, so I wouldn't be. We used to try to avoid the slide scrapings as much as possible because we just didn't see good quality results really at all. But we've completely changed course on those, and now having a reference slide that's marked by a pathologist come in with 10 slides after that will then do the scrapings is a very typical workflow now that works very, very well. Very small cell numbers or spatial type profiling, we have not been attempting that, but we have been thinking about the overall technology on the overall workflow on the Covaris protocol. And if you carefully look at from the tube, the flexibility in the tube sizes all the way down to the bead handling, enabling again, very small cell numbers and even spatial type profiling in a modified version of that workflow should actually be should be pretty well supported through this technology. As I mentioned, DNA yields and quality improvement are not as pronounced as RNA. What we've seen is variable improvements in DNA, really depending on tissue source and type. And I think it just depends on how highly damaged the material was coming in. And so as a final summary, that Covaris is the foundational between their tubes, their instrument, and their technology has clearly demonstrated for us performance for RNA isolations from FFPE material, very enabling from the amount of the diversity of genomic applications that it enables. I also mentioned that microRNA out of those same materials looks really, really good as well. I don't have any data in the slides, but it also looks really quite good. The largest improvement is in the proportion of samples that pass quality control metrics, and we're right now making a deeper evaluation of the improvements in biological resolution. So, it's two different questions. We've shown a drastic improvement in the RNA quality metrics, but it's an open question as to, and we're trying to be very diligent and careful with the analysis to how much resolution does that actually improve in the interpretation of those tumors. There's some clear wins that we've already observed, but I also want to be cautious and conservative without making any broadly sweeping statements that same improvement in sample quality means an equal improvement in resolution of the biological interpretation, because we haven't gotten there yet. So I think it should be an absolutely enabling result. It clearly is enabling in terms of the number of samples that pass QC and go into prep. So that alone, from that perspective, it's biologically enabling. But the question of if you took a sample that was at 30% DV and then you isolated the same sample via the Covaris method and it went to 70, do you see anything new in the data? Right? That's the question that is still under investigation. So last slide, to thank the laboratory team. You'll notice, I didn't notice it until I was working on the slides the other day. Actually, my kids noticed it because they were hanging around when I was working on this talk. That's everybody's head is a little bit misshapen because depending on how their picture was, kind of, cropped to be the same size. So the people that look funny don't actually look that funny in real life, me included. So anyways, this is it's a fantastic team. This is everyone who's currently in the lab. I really need to make a slide that recognizes some of the past contributors as well, both our clinical operation as well as the research operation and informatics. And again, I mentioned my kids. I usually include the kids in the talk because they were complaining I wasn't going to be around this weekend. So I said, okay, you guys can each put a picture in. And so then this is my oldest son's choice was he and his brother got to they rarely play hockey together because of their age difference. They got to be on the same team in a tournament. They won the championship. So this is their picture from that. You may be asking, is this truly an old keg that was used to make a trophy for a 10-year-old tournament? Yes, it is. So you can, this was the Shamrock Shootout, which is here. It was actually a 3 versus 3 or 4 versus 4 tournament. And then my youngest son insisted that I include a picture of his duck. So And the reason I decided to do it, you probably can't quite tell with this here, but this duck is the most judgmental duck. Do you ever see animals that just when they look at you, you swear they're judging you? This duck's name is Elmer, and that's exactly what Elmer does. And he's about, I mean, he's giant. He's about this big. So yeah. Anyways, so with that, happy to take any questions, and I'll put the correct acknowledgement slide back up. Thanks for your attention, and either now or later, happy to take any questions.